Do you know anyone that has been affected by breast cancer? Perhaps someone in your family or friend group. In the United States alone, over 300,000 people are diagnosed with breast cancer each year, and over 80% of those individuals will have invasive cancers. Hello, my name is Caroline Nelson. I am a junior Markey Star Fellow and STAMP Scholar majoring in biology. The research presented here was conducted in part of my continuing work with Dr. Patel in the biology department. 1 in every 8 women will develop breast cancer. Finding an effective treatment could save millions of lives. The Warburg effect describes the cancer phenotype, which is characterized by an increased energy demand and the production of lactate despite being in the presence of sufficient oxygen to break down glucose for oxidative phosphorylation. This process is detailed in Figure 1. The majority of breast cancers are estrogen receptor positive or ER positive, and their proliferation is fueled by the hormone estrogen. ER alpha, along with estrogen-related receptor alpha, ERR alpha, regulate many metabolic genes that promote the cancer phenotype. Tamoxifen, a selective estrogen receptor modulator, has long been a popular drug used in the treatment of breast cancer. However, many patients will develop a resistance to it. The flavanone naringenin is present in many fruits, but predominantly in grapefruit. Naringenin promotes cell death, or apoptosis, and reduces proliferation in breast cancers that are both susceptible and resistant to the estrogen receptor antagonist tamoxifen. The methods through which naringenin works are not fully understood. Our experiments hypothesize that the flavanone produces its effects through further inducing lactate production by altering levels of specific proteins involved in regulating glucose metabolism. This would divert needed resources away from cellular proliferation and survival. Developing an understanding of the mechanism through which naringenin produces its effects is a preliminary step to finding a therapeutic use for the compound in the treatment of breast cancer. MCF7 ER-positive breast cancer cells were cultured until confluent. The cells were then treated with MG132, a proteasome inhibitor, naringenin, or both at various concentrations. Following the Western blot technique, the proteins derived from the cell lysates were separated through sodium dodecyl sulfate polyacrylamide gel electrophoresis, before being transferred onto a membrane incubated with ER-alpha, ERR-alpha, or GLUT4 antibodies. The membranes were then imaged using an enhanced chemiluminescence and biorad-chemi-doc XRS system and quantified using densitometric analysis. Our experiments found that naringenin reduced ER-alpha and ERR-alpha protein levels. The proteasome inhibitor MG132 allowed us to determine how naringenin reduces protein levels. MG132 blocks one of the cell's main degradation pathways, and when combined with naringenin, the treatments revealed that the flavanone prevents MG132-associated increase in protein. This can be observed in Figure 3. Naringenin produced no significant effects in the relative abundance of the insulin-mediated glucose transporter GLUT4, but similarly regulated the effects of MG132 in combined treatments, as seen in Figure 4. Our experiments found that naringenin reduced ER-alpha and ERR-alpha protein levels. The proteasome inhibitor MG132 allowed us to determine how naringenin reduces protein levels. MG132 blocks one of the cell's main degradation pathways, and when combined with naringenin, the treatments revealed that the flavanone prevents MG132-associated increase in protein. This can be observed in Figure 3. Naringenin produced no significant effects in the relative abundance of the insulin-mediated glucose transporter GLUT4, but similarly regulated the effects of MG132 in combined treatments, as seen in Figure 4. Our findings suggest that naringenin is highly selective. Through targeting specific proteins, the flavanone alters protein concentrations in the cell. This effect may be achieved through the activation of the proteasome through a means that bypasses the inhibitor MG132. Alternatively, the proteins may be targeted to the lysosome, another organelle that facilitates the breakdown of cellular materials. These conclusions, as well as that of previous studies, indicate that naringenin has the potential to be a powerful therapeutic agent. Moving forward, our experiments will focus on identifying additional metabolic proteins that are critical in later stages, such as lactate dehydrogenases, to determine the activity of anaerobic processes and further elucidate the mechanisms of naringenin. My name is Caroline Nelson. Thank you for taking the time to view my presentation.